I would like to thank organizers for inviting me to give this keynote lecture at the prestigious 6th World Aging and Rejuvenation Conference in Paris. I only wish that I'm in Paris right now and was able to join in person in this beautiful city, one of the most favorites in the world, and one of my most favorite cities. In this lecture, I will examine evidence of blood-brain barrier dysfunction in the aging brain, particularly in relation to cognitive impairment, Alzheimer's disease, and ApoE4 gene, the major genetic risk for accelerated aging and also Alzheimer's disease. Then I will discuss some brain monogenic diseases when genetic defects are found exclusively in the cells of the vascular system. And then I will also provide examples, discuss some animal models with blood-brain barrier dysfunction that lead to synaptic and neuronal dysfunction, and provide examples of therapeutic approaches directed at the BBB, blood-brain barrier, that can stabilize BBB and slow down neuronal and synaptic dysfunction. Just to remind us of basic physiology of brain circulation, brains receive about 20% of heart output and spends in the resting state 20% of oxygen and glucose. It's only about 2% of body mass, so it's a very highly vascularized and metabolic organ with about 400 miles in human brains of blood vessels. So it's about 600 kilometers. There is this biological membrane that is called blood-brain barrier, discovered like almost 100 years ago, that separates brain from blood compartment and prevents entry into the brain of blood-derived toxic products, cells, and pathogens such as viruses, different bacteria, and fungi. But in addition to preventing and separating brain from blood physically, the blood-brain barrier is a very active metabolic organ of the brain, and is what I like to call it an ecosystem of the brain, like what the ozone layer is for the Earth. It maintains chemical composition in the brain internal milieu necessary for normal neuronal and synaptic functioning, provides all substrates that brain needs for metabolism, and it provides about 10,000 transcripts in brain endothelial cells that are generated there, and about 5,000 proteins, which really shows an amazing high transport activity. Small vessel disease of the brain is found in more than 50% of all dementia, and early blood-brain barrier and vascular dysfunction were found in different diseases, such as Alzheimer's disease in the regions here of hippocampus, ethnocranial cortex, and cortex, Parkinson's disease in the area of substantia nigra, and both corpus caudate nucleus and thalamus are affected in Parkinson's disease and Huntington disease, and the ALS, also amyotrophic lateral sclerosis. It is the spinal cord and motor cortex that are affected, and in multiple sclerosis, also corpus callosum and white matter and the spinal cord. But the question is, we see all these blood-brain barrier changes, increased permeability, leakage of toxin molecule cells, and blood flow reductions as well. And this also is associated with neurological symptoms. But the key question is, what is the role of BBB dysfunction and disruption in cognitive impairment in the aging brain and in relation to dementia and Alzheimer's disease? And are there any treatments that could be acting on a blood-brain barrier that can improve vascular function and slow down neuronal dysfunction and synaptic dysfunction? To address the issue about what's happening in the aging brain and blood-brain barrier, 5 years ago, my collaborator and postdoctoral fellow at that time, Dr. Axel Montagne, who is now reader at the University of Edinburgh in Scotland, we developed this MRI technique, which is magnetic resonance technique that uses contrast agent, gadolinium. It's about 15 minutes essay, and the participants is in the magnet over here. And then we generate individual blood-brain barrier K-trans maps, as you can see it over here. And from this big blood-brain barrier K-trans map, we can calculate entry of this gadolinium to the brain and provide the transfer contents of this entry. And the first observation that we made in 2015 is that in the aging brain, we see in the hippocampus, which is a region of the brain with major role in learning and memory, we see a progressive increase with aging, similar that correlates what has been previously shown with progressive synaptic dysfunction. So here we have an aging effect in individuals between 20 and 90 years old, and not just in hippocampus altogether, but also in a region of hippocampus and subregions, CA1, CA3, and dentae gyrus. What is also interesting when compared age-matched individuals with no cognitive impairment with those with mild cognitive impairment, which is usually classified by UDS system CDR 0.5 and additional neuropsychological test, we can see huge difference here that people who have mild cognitive impairment develop larger blood-brain barrier dysfunction compared to the age-matched individuals that are cognitively unimpaired. And you can here see on hippocampus mask that there is an increase compared to younger individuals in older individuals that are above 65 years old. And also in these individuals that develop mild cognitive impairment, there is even more pronounced blood-brain barrier breakdown. So we published this observation first time in Neuron in 2015 by Dr. Montaigne and our group. And this age-dependent increase in blood-brain barrier permeability that we also called blood-brain barrier breakdown, reported by Montaigne et al. In Neuron for the first time, has been independently reproduced and confirmed by different groups studying different cohorts. And also has been shown that this goes beyond hippocampus and affects all different brain regions. So these are groups listed over here, the Dutch group van der Haar, Verhagen, and then more recently, Bill Yagush group from UCSF has published on this as well. Now, the next logical question was whether these changes in the blood-brain barrier could be attributed to the pathogenic proteins that accumulate in brain with aging, and particularly in Alzheimer's disease, such as amyloid beta protein and tau protein that we know are both vasculotoxic. So we studied patients over here that are divided in those that are A beta negative and A beta positive. A beta negative by CSF and A beta positive, which means do they have amyloid in CSF or in brain or not? And so what these results are showing over here, that regardless whether people are A beta positive or A beta negative, it really the blood-brain barrier follows the cognitive dementia rating score, which is actually clinical score for cognitive impairment. And these individuals that are CDR 0.5, that have mild cognitive impairment compared to those that are CDR 0, would have increased blood-brain barrier permeability in hippocampus or hippocampal gyrus, regardless of A beta positivity, and also regardless of tau positivity. So this was an effect that was independent. And so now we also expanded these studies at the time with the nation who led this study on neuropsychological testing, particularly on memory tests. And he studied all 10 different, 4 domains with 10 different tests, and also found that an increase in blood-brain barrier permeability depends if one plus domain is impaired compared to 0 domain impaired, but does not depend on A beta positivity and negativity or p-tau, Alzheimer's positivity or negativity. So these were reported in Nature Medicine in 2019. And our follow-up study was, does ApoE4, which is the major genetic risk factor for accelerated aging, and the main susceptibility factor for Alzheimer's disease, does ApoE4 has any effect on blood-brain barrier permeability? So we have in our body 2 ApoE alleles, and there are 2 ApoE genes, but there are 3 different ApoE alleles: ApoE2, which is protective; ApoE3, which is average risk for Alzheimer's disease; and ApoE4. And ApoE4, there are 5% of individuals, increases risk for Alzheimer's disease by 15 times and cognitive decline. But those individuals that are ApoE3 and 4, that has 1 protective allele and 1 bad allele, it's 18% of them, still the increase is about 3-fold compared to the average risk. So then this is a lipid carrier, you know, ApoE, so it's related to lipid metabolism, and we were really interested to find out now what is the effect on blood-brain barrier in individuals that are carriers of ApoE4 compared to individuals that are non-carriers and that are homozygous to have both ApoE3 genes. So this first study that was published in Nature, journal Nature, it was led by Axel Montaigne, and some statistical analysis that were performed by Dr. Danation. We showed that actually in CDR stage 0, which is apparently cognitively unimpaired person, there is an increase in blood-brain barrier permeability in people who are ApoE4 carrier compared to those who are ApoE3. And this increase is further accelerated when the stage progresses to CDR stage 0.5, which is mild cognitive impairment. At that stage also, ApoE3 carriers show an increase in blood-brain barrier permeability. And this was shown over here when Dr. Montaigne computed this K-trans constant. You can see that there is an increase in an initial stage, but there is also, importantly, there is an increase between CDR 0 and CDR 0.5 over here, as shown, in the red in ApoE4 carrier, but also in ApoE3 carrier, but is not so prominent. Again, this increase was independent of A beta pathology or tau pathology in CSF, as shown by this analysis. Now, to be sure about this independence and independent pathways, we correlated this data in these patients, in these participants actually, using amyloid PET, which molecular imaging of amyloid in brain, and tau PET, which is molecular imaging of tau in the brain. And so we performed this imaging and also in the same time K-trans constant. And so the guy who did this imaging, Arat Chakoyan, on amyloid PET and tau PET, and Axel did Montaigne on K-transes. They show that there is a small increase, but not significant, between E3 and E4 carriers at that stage in amyloid accumulation, and also a small increase in that stage in tau accumulation in the hippocampus. But there was no difference between ApoE4 carriers and ApoE3 carriers. While at the same time, the finding that there is an increased blood-brain barrier permeability or breakdown in the hippocampus was also shown here in the same patients, indicating that this breakdown is not related to increases in tau or increases in amyloid, confirming basically much larger data set in the CSF samples that was using 256 participants. But then in the regions that develop amyloid accumulation, such as orbital frontal cortex, we confirmed an increase in amyloid in ApoE4 carrier, which is one of the initial findings in this ApoE4 carrier in terms of amyloid pathology. But there was no difference in blood-brain barrier permeability, and the K-trans constants were the same. That also confirmed independently that there are A beta and tau status in brain is independent on blood-brain barrier breakdown. And then blood-brain barrier breakdown develops both in individuals that are in Alzheimer's path and who will eventually develop Alzheimer's disease that are positive for amyloid beta and tau, as well as individuals who are negative. So maybe they're in early stage, they will develop, or maybe they will never develop and probably will remain with vascular dementia. So another interesting thing, and this was work performed by Giuseppe Barisano, who was also first author in this paper, was that hippocampal volume was, of course, reduced in ApoE4 carrier that are cognitively impaired with 0.5 compared to those that are ApoE4 0. But interestingly, this finding was basically shown, the blood-brain barrier disruption was shown in these ApoE4 carriers, even that they don't have the generation of hippocampus before the generation of hippocampus is found, as shown over here. So that the generation practically followed changes in the blood-brain barrier permeability or increased changes in blood-brain barrier permeability. So this work was published in Nature, and ApoE4 leads to blood-brain barrier dysfunction, predicting cognitive decline, and findings that ApoE4 leads to this has been shown first by Montaigne, have been independently reproduced recently by different groups, by 3 different groups over here. Now, blood-brain barrier breakdown has been shown in Alzheimer's disease by pathological studies. There are more than 20 postmortem studies that have shown this previously before we start doing imaging. And there is accumulation of fibrinogen, which is blood product in the brain, very toxic, that obviously colocalizes with amyloid in the brain. But also in this electron micrographs that was provided by the Greek group by Alanis, you can see there is accumulation of osmophilic particles in the pericytes, and pericytes are engaged into absorbing a lot of this material that comes from blood, trying to prevent it to come into the brain, and then they eventually degenerate, and also the endothelial cells degenerate. So we were very interested in pericytes because there were several studies showing that pericytes indeed maintain the blood-brain barrier integrity, in addition to this clearance function of toxins, and also neurotrophic functions that some of my colleagues have shown, and also role information of new blood vessels. So Dr. Segare and my PhD student at the time, Melanie Sweeney, were working on some biomarkers, and they actually discovered that soluble form of PDGFR-beta receptor, which is characteristic receptor for mural cells, both pericytes and vascular smooth muscle cells, is released mainly from pericytes into the CSF, and is mainly released from pericytes by ADAM10 enzyme, which degrades PDGFR-beta and makes soluble form, and also ADAM17 is involved. And these enzymes were not so much expressed in vascular smooth muscle cells. So then that was the reason why PDGFR-beta was actually coming most likely from pericytes and was assumed to be a pericyte marker. So we first reported this in paper by Montaigne, when Segare provided this data, showing that MCI participants have much higher levels of this peptide than actually people who are cognitively unimpaired. And interestingly, this increased CSF levels of PDGFR-beta correlated with increased levels of blood-brain barrier permeability in the hippocampus and some of the hippocampal regions. So this was really very nice match. So in the larger cohort that included participants from USC and WashU, we confirmed this data. The nation confirmed this data, and Segare provided this as PDGFR-beta levels. And people with 0.5, with mild dementia, with mild cognitive impairment, have showing increased levels, and this was more increased when the levels are actually maintained, accelerating in the CSF. And we were interested to see whether this was influenced by A-beta 42 levels, which is downregulated in these patients in the CSF, or tau levels, which are upregulated, that are elevated. And so what we were able to show that after corrections for A-beta levels and tau levels, and stratifying people that are A-beta negative, tau, A-beta positive, and tau negative and tau positive, we did not find that A-beta pathology was influences as PDGFR-beta levels, similar as with the K-trans. So this was completely a novel independent pathway. So after this discovery of PDGFR-beta, different groups studied PDGFR-beta and that reported first in Montaigne. And for example, it was shown that PDGFR-beta increase in AD compared to age-matched controls correlate positively with dysfunction of the renin-angiotensin system, reflecting capillary damage, is increase in preclinical stage of AD. It is also increase in preclinical stage GDR-0.5, shown by other group, and et cetera, et cetera. And there were also studies showing correlation between PDGFR-beta and tau and beta 40 pathology, showing that cerebral vascular dysregulation may be a very early event that occurs in the development of AD. So that has been shown by several groups. Now, in our original paper in Nature, the nation was asking this question: can high pericyte injury at baseline, indicated by highest PDGFR-beta levels, can it predict cognitive decline in APOE4 carriers? And is it related to APOE genetic risk? So he studied 350 participants for median split. Those are people above median and below median. And he showed by follow-up longitudinal studies for 2.5 and 4.5 years that people who have higher levels progressively deteriorate on mental status test and global cognition test, not shown here, which is not same for individuals that are APOE3. So there was much more sensitivity in APOE4 individuals to this pericyte injury. Most recently, Dr. Segare generated data on plasma PDGFR-beta, and as shown here, confirmed our finding in CSF that these levels are increased in APOE4 carriers compared to APOE4 carriers at CDR.5's MCI stage compared to 0 stage. And Dr. Nation then provided longitudinal analysis of this, corrected for age, sex, education, and vascular factors, showing that people who have higher levels than 84 nanogram per ml of sp-GFR beta decline much more rapidly comparing to those with lower levels. And then he also did mediation analysis and showed that sp-GFR beta high levels that directly influences cognitive impairment at CDR.5 and also can act on influencing the volume, hippocampal volume degeneration as well. And then hippocampal volume degeneration would act also on cognitive impairment. So that's a double pathway. So the group who studied NIA, AIA revised clinical criteria included sp-GFR beta as a microvascular pathology biomarker. So that could be studied in parallel with A beta pathology, neuroimmune, and inflammatory system pathology, tau pathology, and synaptic dysfunction and axonal damage as an important parameters. Now, since this is rejuvenation conference also, I want to say that we've done some work with the stem cells, and we generated iPSC-derived pericytes, which are these cells that maintain the blood-brain barrier. And so they're generated by process via neural crest cells. And when we compare these pericytes with primary pericytes using proteomic analysis, we found actually Dr. Kova's lab and Veronica and also Russell and Rask were working on this. They found that 96 proteins are expressed quantitatively in similar levels in these stem cell-derived pericytes compared to naturally occurring pericytes isolated from brain. And that phosphorylation sites in 98% are identical in this between these two pericytes. And so then we did analysis of different pericytes biocanonical markers. We showed they're exactly the same in both groups. Cell adhesion proteins were 100% same as well as their phosphorylation, and also tight junction proteins were 100% same and as well in both pericytes and iPSC derived as well as their phosphorylation. Regarding transcription factor, there was about 96% of transcription factors are equally expressed or similarly expressed comparable levels in these pericytes and 96% of phosphorylation sites. And similar with protein kinases, which actually regulate signaling in cells, there are similar, about 94% were comparable and 98% in phosphorylation site. So when Dr. Kova did this analysis of human kinome, he confirmed high level of similarity in protein kinase levels and also high level of similarity in phosphorylation. So these are still unpublished data. Now, the functional assays that were performed here on isolated slices with my former postdoctoral fellow, Amy Nelson. Over here, we still have this data, but we are now working on generating much more data to show that these pericytes, when they're placed on slices, on mouse slices, on pericyte deficient mice, they actually go and form hybrid vessels. But the pericyte receptor, PGFR beta, is knocked down. They cannot do that, and they remained rounded. And similar was shown in the brain after intrahippocampal injections that was shown by Dr. Wang and Dr. Montaigne. Now, Dr. Segari and Alison Bosworth performed Claudin-5 and Z01 staining. These are tight junction proteins. And as you can see, the pericyte media are improving much, this network that actually, keep junction endothelial cells together, both shown with Z01 also here. So improved BB integrity in vivo was shown by MRI in this F7 pericyte deficient mice that received iPSC-derived pericytes compared to controls. They don't receive, so the higher the color here, these are MRI maps that were provided by Dr. Chakoyan and work by Dr. Montaigne and Chakoyan, is showing that really these pericytes are closing the blood-brain barrier in vivo. And when you stop, and when you silence this receptor PGFR beta, they're losing that property. So now the BBB leaks lead to neuronal and synaptic dysfunction, injury due to degeneration. It's shown by many studies. And there are a number of molecules such as red blood cells and toxic iron species, such as fibrinogen that activates microglia, such as thrombin that is directly neuron toxic, and accumulation of plasmin and also albumin that activates astrocytes and formation of antibody. That all happens when the barrier is open, as well as loss of neurotrophic support. But there are rare human monogenic brain diseases and animal studies that further support this connection. For example, these dozen diseases, genetic defects are found at the blood-brain barrier or blood-brain barrier-associated vascular cells, not anywhere else in the brain. And then these individuals develop serious neurological diseases. This was found in GLUT1 deficiency syndrome, and also MSF, D2A missense, mutations in endothelial cells, where you have microcephaly, intellectual disability, speech disability, and many other disorders that are listed over here. So these disorders are the best evidence for the causal pathogenic link when the BBB dysfunction leads to neurologic disease. Now, some of the work from Little Winkler, my former PhD student, that shows over here, is that animals that are lacking GLUT1 transporter, similar to this human GLUT1 deficiency disease, have low uptake of glucose. This glucose transporter is exclusively in brain endothelial cell. This leads to leakage of the blood-brain barrier very early. And then after 6 months, you have functional problems in neurons. That's shown by image of all cortical membrane potential responses by slower, time when the neurons fire, and also by lower peak of firing. In addition to that, there was 4% microcephaly, which is smaller brain already at 6 months. And that's only in the presence of that transporter lack. In the absence of this transporter, similars were shown with transport with essential omega-3 fatty acid by lab of Chen-Hua Gu. And there is a blood-brain barrier knockdown in these mouse that lack this gene and maintains blood-brain barrier integrity, and is followed by neurodegeneration microcephaly. Another gene that we studied is lipoprotein receptor. Its loss also leads to actually blood-brain barrier breakdowns, as you can see here. But neuronal loss is only happening later at 4 months of age. What was very interesting when we treated with blood-brain barrier gene therapy, when we put back this gene in about 50% of the vessels that's shown here, this partially improved blood-brain barrier integrity and also slowed down neuronal degeneration that remained, in this case, normal in animals that were receiving this blood-brain barrier gene therapy. Now, I want also to provide one more example, which is cyclophilin A and MMP9 pathway, the discovery in Nature 2012 paper in animals and also confirmed in humans in Nature 2020. This is a pericyte, activation of pericyte pathway that leads to blood-brain barrier degeneration by MMP9 degradation of tight junctions and collagen, and it's related to increase of cyclophilin A. So the inhibitors that actually inhibit that cyclophilin A were able to recover the voltage-sensitive dye curve of cortical activation responses to kind limb stimulation in these animals. So time to peak has been reduced, and also the amplitude of peak has been increased as well, and increased expression of postsynaptic and presynaptic proteins just by protecting this blood-brain barrier. So the key points to take message, and I will finish shortly, is that BBB breakdown is an early biomarker in the aging human brain and the associated cognitive dysfunction. APOE4 carriers compared to APOE3 homozygote develop preclinical BBB breakdown in the hippocampus and medial temporal lobe, which is more severe with cognitive impairment, but is not dependent on A-beta and tau Alzheimer's pathology. Elevated SPGF-beta CSF plasma levels can predict cognitive decline in APOE4 carriers. BBB dysfunction in transporter genetic defects leads to CNS dysfunction, like in human monogenic brain disorders. And then I provided therapeutic approaches that show directed at DBB can improve vascular function and slow down neuronal dysfunction. The examples of cell therapy with iPSC-derived pericyte or gene therapy with brain endothelial AAV gene delivery of LRP1 and small molecules inhibitor of CPA MMP9 pathway. I would like to thank for my past research support, the NIH, NIA and NDS, Alzheimer's Association, Cure Alzheimer's Fund, the Duck and Open Philanthropy, and the talented group of researchers in my group that are amazing and actually that are performing all this wonderful work. Thank you very much for your attention. Open for question.